to Doctor and everyone. My name is Wan Izzat Atira Pituan Kamaruzaman, metric number D201910888446. Okay, our experiment was titled Characterization of Organic Matter of Plants from Lakes using Thermal Analysis in Nitrogen Atmosphere. In this part of video, I will explain three things, which is the first one is objective, introduction, and the last one is methodology. Okay, for the first part is the objective of this experiment. Okay, there are four objectives in this experiment. The first one is to identify whether the characteristic peak at 300 degrees is a shoulder peak generated from two superposed exothermic peaks or a transition stage between an endothermic peak and an exothermic peak. Next is to identify whether the characteristic peak at 460 degrees is an endothermic peak or a transition stage between two exothermic peaks. Next, to determine the relationship between peaks of organic matter at different stages and their components. And the last one is to determine the relationship between characteristic peaks of organic matter at different stages. Okay, next part is introduction. Organic matter is important as complexing agents or absorbent of environmental pollutants found in surface water. However, conventional methods of characterizing organic matter are unsatisfactory because they do not capture the complete continuum of behavior of organic matter. So, thermal methods, which have simple pretreatment and short experimental time, have been shown to be easy, fast, relatively inexpensive, yet reliable for characterizing organic matter. Moreover, thermal analysis has been demonstrated to be useful for identification of type of vegetation from which organic matters in soil and sediment have derived. Lastly is methodology. Okay, so in this experiment, there are three methods involved. The first method is preparation of sample. 17 plant sample is collected from the lake, consists of four categories, which is emergent plants, submerged plants, floating plants, and terrestrial plants. Next, plant sample were dried and ground passed through to sieve to make it as powder. 8 standard sample is used in this experiment to make comparison with the result. The 8 standard sample is glucose, xylose, D-arabinose, L-arabinose, L-rhamnose, D-galactose, cellulose, and lignin. And the last process in this method is using humic acid to verify the result. The second method is thermogravimetric analysis. This method is using TGA Q50 analyzer. This method is to evaluate thermal stability of sample and evolution of physics of water. The last method is differential scanning calorimetry. This method using DSC model Q20. Okay, to improve the accuracy of DSC, we weigh the samples and reference to ensure that difference between both sample and reference less than 0.1 mg. Next, samples are placed into a standard aluminum pan under an atmosphere of nitrogen gas. After that, each sample pan is conditioned to remove the physics of water. DSC is conducted while heating from ambient to 600 degrees at 10 minutes. Next, total organic carbon (TOC) is measured by use of total organic carbon analyzer by the non-dispersible infrared absorption method. The last one is thermal indices such as peak area and peak temperature of each peak is determined by Universal Analysis 2000 software.
Hi, my name is Joeria, and today I am going to present about the result and the conclusion for this experiment.
Next is the result. The peak is observed from the differences in peak's behavior, area, and also height. Firstly, for the result of differential scanning calorimetry analysis. Figure 1 shows the differential scanning calorimetry scans of D-arabinose, cellulose, and lignin, while Figure B shows the D-acid scans of HA, which is the humic acid from sigma and plant 5 from Thankyou lake. For the monosaccharide D-acid curves, which is the D-arabinose, there are 4 different peaks observed, which is 3 endothermic peaks at 159 to 170 Celsius, 207 to 222 Celsius, and 292 to 312 Celsius, as well as an exothermic peak at 305 to 343 Celsius. For the cellulose, there were only 2 peaks observed, which is an endothermic peak at 336.2 Celsius and an exothermic peak at 378.6 Celsius. Lastly, for the lignin, exhibited 2 endothermic peaks at 205.4 Celsius and 278.0 Celsius, and also an exothermic peak at 384.4 Celsius. Hence, based on the figure, we can conclude that the first endothermic peak appeared between 93 to 110 Celsius. This peak was caused by the melting or glass transition of small molecules. The second endothermic peak, F2, occurred at 175 to 217 Celsius. The first endothermic peak of lignin and the main endothermic peak of monosaccharide occurred at almost the same temperature, which is at 205 Celsius. Therefore, the F2 peaks observed were likely the superposition of several thermal processes and cannot be individually characterized based on the data generated in this experiment. As we can see here, a third endothermic peak was observed at 277 to 311 Celsius in every scan of plant materials, while another endothermic peak near F3 was observed between 344 and 358 Celsius. In this temperature range, scans of monosaccharide show that the end of the first endothermic peak, while scans of cellulose were in the process of heat absorption, which is 30 Celsius before its endothermic peak. This temperature was between the end of the second endothermic peak and the exothermic peak of the scan of lignin. Next, based on the figure, the fourth endothermic peak was observed at 404 to 437 Celsius in some of the scans. A remarkable exothermic peak, which is the Fx, occurred in the range of 305 to 450 Celsius. Peak Fx represent the main exothermic process required or suggested by the corresponding thermal behavior of all standard samples. Lastly, an endothermic peak, which is the F5, occurring among it in the range of 444 to 480 Celsius. Lignin was the only standard material showing corresponding peaks in D-acid scans, while monosaccharide and cellulose remain stable in this temperature range. To conclude, F5 peaks in lignin scans behave like transitional peaks between two exothermic processes. Next, we have the thermal gravimetric analysis. Figure 2 shows the DSC and DTG overlay analysis of lignin. It is important to verify if degradation occurred in DSC peaks and confirm the thermal significance of each DSC peak. DSC and DTG curve were superposed and analyzed. The DSC and DTG relationship is the foundation of plant biomass behavior analysis. To conclude, for the monosaccharide, endothermic peak can be observed at 210 Celsius with a loss of mass peak at 230 Celsius. For the exothermic peak, the peak was observed at 330 Celsius with a loss of mass peak at 340 Celsius. Then, a slow heat releasing and slow loss of mass after 400 Celsius. For the cellulose, endothermic peak was observed greater than 335 Celsius, then rapidly enters an exothermic stage, peaking at 378 Celsius, with a loss of mass peak starting at 380 Celsius and ending at 450 Celsius. Lastly, to conclude for the lignin, there are endothermic peak observed at 205 Celsius with a loss of mass peak at 232 Celsius, while a smaller endothermic peak was observed at 277 Celsius, followed by a strong exothermic peak. Additionally, strong loss of mass occurs at 343 Celsius, while exothermic processes peak later when the rate of lost mass decreases. Next, Figure 3 shows the lag relationship of peak F2 and peak Fx in DSC and DTG scans of standard samples. To conclude, there was no obvious loss of mass peak and a lesser rate of loss of mass in F1 peak at 93 to 110 Celsius, suggesting that peak F1 represents a melting process, which is consistent with the DSC result. Peak F2 represents a recombination process of endothermic melting in the first half and endothermic degradation in the latter half, while peak F3 represents a stable stage before the strong exothermic degradation, but not an endothermic loss of mass. Lastly, most samples with peak F5 showed larger loss of mass peaks at 460 Celsius, coinciding with the behavior of lignin.
Assalamualaikum warahmatullahi wabarakatuh. My name is Wanurul Fakihah binti Wan Hamdi, and today I will presenting about the discussion of this lab report. The peak areas of F2 and TOC were well associated, and the correlation coefficient R of the peak area with P equal to 0 of extracellulose, hemicellulose, and lignin. This result show that peak F2 is a combination of organic materials. Peak F2 never appeared to be related to any composition. Overall, as well as minor decomposition, the processes resulting in the F2 peak are predominantly dominated by absorbing energy. With each component and total organic matter, the absorbed heat of peak F3 was poorly associated, indicating that peak F3 was not endothermic decomposing peak but rather a smooth transition time. Peak Fx represents the main exothermic process suggested by the corresponding thermal behavior of all standard samples. Peak F5 was observed in the middle of the Fx peak. Instead of endothermic pyrolysis peak, peak F5 reflects the difference between endothermic processes. Nevertheless, this F5 peak can be used to characterize lignin content. Based on all experimental findings, STDN of plant biomass could be divided into three stages. Peak F2 represents endothermic primary degradation processes that occur at a relatively slow rate. This was followed by peak F3, reflecting a transitional stage in which energy was still being absorbed, while the Fx peak represents severe exothermic activity and rapid loss of mass. This study provides a foundation for understanding the biomass STDN. Results reveal that several thermal behaviors overlap, making it difficult to identify component in organic matter. Slow thermal degradation in an N2 atmosphere could be a potential method for characterization and even identification of organic matter.
Next is the conclusion. There are four conclusions The first one is the characteristic peak at 300 Celsius is a transition stage between an endothermic peak and an exothermic peak. The second conclusion is the characteristic peak at 460 Celsius is a transition stage between two exothermic peaks. The third one, the relationship between peaks of OM at different stages and their components is determined. While the last conclusion is that the relationship between characteristic peaks of organic matter at different stages is also determined.